How do prokaryotes and eukaryotes differ for med school? Imagine trying to understand the tiny building blocks of life. Some are simple, while others are more complex. This difference is key in medical microbiology and studying how germs and human cells work. When you start your first or second year of medical school, knowing how these cells differ helps you grasp how infections happen and how medicines target bacteria without harming us. Prokaryotes are the simplest cells. They do not have a nucleus, which is like a control center for DNA. Instead, their DNA floats freely in a region called the nucleoid. Their DNA is a single circular piece, unlike our linear chromosomes. They do not have histone proteins, which help organize DNA in human cells. Because of this, their genetic material is packed differently. Prokaryotic cells are also smaller, usually between 0.1 and 5 micrometers. They lack membrane-bound organelles like mitochondria or endoplasmic reticulum. Instead, energy is produced across their cell membrane. Their cell wall is often made of peptidoglycan, a complex molecule that antibiotics like penicillin target. Eukaryotic cells are larger, ranging from 10 to 100 micrometers. They have a true nucleus enclosed by a nuclear membrane, which keeps their DNA separate. Their chromosomes are linear and associated with histone proteins, helping regulate gene activity. These cells contain many organelles such as mitochondria for energy, endoplasmic reticulum for making proteins and lipids, and lysosomes for digestion. The cell walls of eukaryotes vary. Plants have cellulose, fungi have chitin, but animals do not have cell walls at all. Ribosomes in eukaryotic cells are bigger, about 80 Svedberg units, while prokaryotic ribosomes are smaller, around 70 Svedberg units. This difference is important because some antibiotics specifically target bacterial ribosomes, leaving human ribosomes unaffected. When it comes to reproduction, prokaryotes divide quickly through binary fission, a simple process without the need for mitosis. Eukaryotic cells reproduce through mitosis for growth and repair, and meiosis for making reproductive cells. This makes eukaryotic reproduction more complex and slower. Prokaryotes often carry plasmids, small circular DNA molecules separate from their main chromosome. These plasmids can carry genes for antibiotic resistance and can be transferred between bacteria. Eukaryotic cells rarely have plasmids. Lastly, motility structures differ. Prokaryotic flagella are smaller and have a different structure than eukaryotic flagella, which are larger and have a 9+2 arrangement of microtubules. Understanding these differences is vital in medical school because it explains how antibiotics work and why they target bacteria without harming human cells. It also helps you see how bacteria adapt and resist drugs, which is a big part of infectious disease studies. Knowing how cells are built and how they divide makes it easier to understand disease processes and develop treatments. So, in your first and second years, focus on these fundamental differences to build a strong foundation in medical microbiology.